given all the exclusion criteria and randomized controlled trials, sometimes you have to wonder just exactly how well their cohorts really reflect the standard patient population. So towards this end, we want to talk about an interesting paper being presented here at ASH 2015. I'm with Abby Statler, who is an MPH, an MA, and a PhD candidate. She is the Research Regulatory Quality Assurance Coordinator at the Cleveland Clinic in Ohio. And we're talking eligibility criteria are not associated with expected or observed adverse events in RCTs of hematologic malignancies. What was the impetus behind this very interesting study, first off?
Yeah, so I think we have this push and pull between internal validity and external validity. And when I say internal validity, I'm talking about a trial's ability to demonstrate a cause-effect relationship. And when I say external validity, I'm talking about the actual generalizability of those results that are presented on those trials. And it's a challenge to balance these two weights. And when I think about this, I think it's important to ask the question: are we compromising exclusion or external validity by focusing on internal validity? And a way to look at that is to say, are eligibility criteria too strict? And then that means that we are not able to generalize the results from these trials to the general population.
Just what I said at the open, it's a real challenge.
Yeah.
So how did you do this study?
So we pulled randomized controlled trials in hematologic malignancies from medical journals with an impact factor of 5 or greater, and we pulled those studies and we extracted the data relevant to the eligibility criteria as well as the toxicities reported on those studies. And then we compared the number of studies that we expected would report toxicities relevant to their exclusion criteria to the number of studies that actually reported those toxicities. And then in addition to that, we also looked at the percent of drug classes with known toxicities, and we compared that to the number of trials that excluded patients with the eligibility, with the exclusion that would be relevant to that toxicity at baseline.
I suppose it's fair to ask, what were your exclusion criteria?
Sure. So our inclusion criteria were, I'm going to focus on inclusion, were randomized controlled trials, phase 2 or phase 3, hematologic malignancies, adults, and then published in the last 5 years, and they had to be a therapeutic intervention.
Okay.
Yep.
And in terms of exclusion criteria, you didn't really have any. You were just looking at the.
So if it was not an RCT, we didn't include it. So that,
right.
I guess you could say the opposite of our inclusion criteria was our exclusion criteria, essentially. Yeah.
Okay. So what did you find?
We looked at 4 different areas of exclusion criteria. So we looked at hepatic exclusion criteria, renal exclusion criteria, cardiac, and then neurological. And so what we found, relevant to the expected adverse events, is that 75% of the major drug classes that treat hematologic malignancies actually have hepatic toxicities. But 87% of our studies excluded patients with hepatic abnormalities at baseline, which was a big difference with a significant p-value. The same followed for renal. So we had renal, we had 50% of drug classes have known renal toxicities, and we identified that 73% of our studies actually excluded patients with renal abnormalities at baseline. So again, a big difference and a significant p-value there as well. So when we look at the observed adverse events, we say we anticipated so for instance, looking at hepatic toxicities, we expected of the 85 studies that excluded patients with hepatic toxicities at baseline, we expected that 41 of those studies would actually exclude patients or would actually report hepatic toxicities, and we only saw 19 studies actually report hepatic toxicities. And this same trend followed for renal and cardiac. Neurological was different. We anticipated that 11 studies would report toxicities, and actually 14 reported toxicities. So that was higher than we anticipated. Yeah, and yeah, those are really our major findings.
So you have a graphic showing expected versus observed adverse events that is quite striking.
Yeah.
Were you kind of surprised by what you found?
We were certainly surprised by. I think our hypothesis was that we would see. We would have fewer studies report toxicities than we expected, but I don't think we anticipated that the difference would be that dramatic. For instance, when you think about the hepatic toxicity exclusion criteria, we expected 41 studies to report hepatic toxicities, and we only saw 19 report those, which is a huge difference.
Why? What was driving this?
So what may be driving this is the inclusion criteria and the exclusion criteria. So that's to say that we are enrolling patients that are very clinically fit, and so they don't have a lot of toxicities. And so what happens is we have this excellent patient population that is very well positioned to potentially present a clinically meaningful and statistically significant result, which then results in very good safety data, which may not actually be reflective of what happens on a daily basis in your clinic. And so the safety data is actually really good on these studies. And what we're arguing is that we may be able to expand exclusion criteria a little bit to include more patients, and we would not significantly impact the safety of the actual investigational product.
And that way you get a better idea of what's going to happen in actual clinical practice.
Absolutely, yeah. So the whole idea is the fact that once you get to phase 3, randomized controlled trials, you're looking at efficacy. So you've really identified the safety profile. So at this point, it might be appropriate to include more patients to identify the efficacy of these agents. And in that way, once the drug is on the market and it's actually utilized in the general population, we understand how it works in patients that have comorbidities, how it works in patients who have a minor organ function abnormality. And I think that's the most important thing about this particular study. In addition to that, I think that what's really interesting is that cancer is a specific disease that's life-threatening, as we all know. And so sometimes clinical trials are a patient's last option. Yeah. And so by excluding these patients at baseline, we're essentially denying them access to novel therapies, which might actually be their last option. And I think we need to reconsider that. We need to think about how we design eligibility criteria and be more purposeful about it so that we can increase the generalizability of the results and then provide access to patients who might not have access otherwise.
I mean, when you have so much money invested in a clinical trial, you want to make sure that you get not just a good result, but that you get an accurate result with these people that you have enrolled. And
Absolutely.
So there's this nervousness of enrolling these extra people.
Yep.
You think there's a way to balance it, though?
Absolutely. And so I don't think that, from a patient safety perspective, I don't think that we should, include patients that have, severe kidney disease or renal failure or severe cardiac condition. But those patients who may have a non-clinically significant comorbidity, why can't we include them on the clinical trial? Is that really going to affect the efficacy? And I think that many people in the field would argue probably not.
So do you think your take-home message here is let's rethink this at the very start of a clinical trial?
So let's think about our eligibility criteria, and let's design eligibility criteria that is specifically informed by the expected toxicities based upon the drugs that are being investigated. So we know once we get to phase 3, we know that we have a general sense of the toxicity profile of those agents. So why don't we look at the eligibility criteria and design the criteria based upon that knowledge that we already have? I think sometimes we are reflexive about using eligibility criteria across many different studies, and this is not well thought out. And by doing that, we're excluding a large group of patients, specifically those with comorbidities, and then we're also denying patients access to clinical trials. I mean, at the Cleveland Clinic, we see this, regularly, where we have a patient who comes through our doors and we really want to put them on the clinical trial, but we can't because they have a minor comorbidity. They had, a cardiac condition that is not clinically significant. Their bilirubin is just a little bit increased, but that means that they can't be enrolled. And especially if you're traveling from a far distance and you have to be rescreened, that patient may not come back to be rescreened for that clinical trial.
I mean, there's something to be said about consistency, but on the other hand, let's start something new and get a new consistency going with a little bit wider open.
Absolutely. And I think right
criteria.
now the federal government is actually is emphasizing what's called pragmatic clinical trials, which are studies that are designed in a way that hopefully produce results that are more generalizable to the patient population. So again, focusing on patient and patient outcomes versus really focusing on producing a perfect drug, really producing a drug that's actually effective in the patient population. And those trials are, they actually do employ eligibility criteria that may be a little looser. And they say, the FDA has said that these eligibility criteria can be loosened depending upon the context of the randomized controlled trial. And not to say that we include all patients, but that you purposely design those clinical trials so that you can enroll a patient population that's reflective of the patients who walk through your doors on a daily basis.
I think it's a great idea. Thank you very much. I appreciate your time.
Thank
And we have all kinds of reporting from ASH Clinical News, so please look around online and, of course, in print. I'm Rick McGuire.